Good afternoon, everybody. First of all, thank you for coming and on such a gorgeous day out there. This is a pretty special day because this is graduation for the folks in the second row, not the folks in the first row. You've already graduated. The folks in the second row who are graduating from an NIH-funded research experience integrated with the clinical experience. And I'm sort of jealous because, I mean, to earn your first NIH grant when you're under 21, it just seems unfair, okay? There's just something really wonderful about that. And I think you all know how special you are, not just because your parents have already told you that 1,000 times, but also that you competed to get this grant. And then every scientist in every lab I talked to could not be more complimentary on the intellect and the enthusiasm and the energy that you brought to this program. So remember, the thing I asked you to do after this program is to pay it forward. All we want you to do is to live up to your best, to live up to your potential. We love when we hear people have gone off and become neuroscientists or scientists of any kind, become physicians, nurses, physician assistants, whatever it is that you dream of being in your life. I hope that in some way this inspired you to go further. So I want to thank you. But before we start with the talks, and I know you're all nervous, you're going to do brilliantly, don't worry about it, okay, I first want to thank the administrative staff in here. And when I say administrative, they're the scientific staff. So the director of research is sitting right over there, Dr. Christine McDonough, okay. You have her mostly to thank because she runs the selection process. I have recused myself from that, and she brilliantly does that. Every year I say, Christine, they just keep getting better. I don't know where you find these young people, and you're the best year, okay, so far. Okay, I say that. All right, Julie Bold over there, our administrator. Jim Pridgeon, who was one of the creators of this program with my wife and I, and Jim has been very much the soul of this program. And then, of course, Sylvia, who is our educational director for the Department of Neurological Surgery, who we could not do this without her. So Jim and Sylvia, thank you so much. The soul and the hard work right here. Now, I want the second row to stand up and applaud for all the scientists who actually had you in their laboratory. And while you're standing, thank your parents. Some of the parents are here. Thank your parents and thank all the people that helped get you here. Okay. So parents, thank you for producing these children, and now grown-ups, and remember, pay it forward. Okay, so thank you. Why don't we all sit down, and we're gonna what the format of this is the students going to get up and talk about their research. And I want to, this is part of the program. Part of the program is it culminates in this wonderful talk that they're going to give. So without further ado, I want to introduce Carolyn Kwan, who's a rising senior at the University of Washington, and she worked in the Davis Neurology Lab at the VA. So Carolyn, and you have your parents now. Why is it working? Yeah, it just froze up because we're giving the talks. Okay. There you go. Okay.
There we go.
Are you gonna be okay like that?
It's a little tall.
It's a little tall. You want me to
No,
you
no,
can
no.
stand on my back if you want.
Thank you. Hello, my name is Caroline Kwan, and I had the privilege of working at the Davis Lab at the VA hospital this summer. And today I'm just going to be presenting a little bit about the research I was able to do about the mechanisms underlying Parkinson's disease and the genetic risk factor GBA. So just as a bit of background on Parkinson's, it's the second most common neurodegenerative disease after Alzheimer's. It affects 1 to 2% of the population over 65. Some common symptoms include tremors, postural instability, rigidity, cognitive decay, stuff like that. And it's also characterized by the presence of Lewy bodies, which are these abnormal aggregates of including ubiquitinated proteins and also this specific protein called alpha-synuclein, which I'll get into. And interestingly, majority of Parkinson cases are actually sporadic, which have no clear genetic or environmental cause. So the spectrum of genetic influences on Parkinson's is actually very complex, and it can range from these rare single causative genes with almost 100% penetrance to very common but clinically insignificant genes. And the gene we look at, GBA, is very interesting in that it's sort of right in the middle. It has clinically significant penetrance. It increases the risk of developing Parkinson's by about 5-fold, and it's present in 5 to 10% of the population. GBA encodes a lysosomal enzyme, and it's known that if you have a recessive mutation in GBA, you actually, it causes Gaucher's disease, which is the most common lysosomal storage disease. But heterozygous GBA mutations are the strongest genetic risk factor for Parkinson's. Like I said, it increases by 5-fold. And GBA carriers with Parkinson's have more rapid disease progression than Parkinson's patients who are non-carriers. So normally when proteins are targeted for lysosome degradation, they're tagged with ubiquitin. In the case of a GBA deficiency, these ubiquitinated proteins sort of aggregate and are unable to be degraded, leading to Lewy pathology. As we can see by the scary red bar, when a fruit fly has a GBA deficiency, the levels of ubiquitin are very, very high. And we can also see, as indicated in the green, that when we reintroduce the GBA into those fruit flies, there's actually a rescue of that aggregation. Previous work in the lab also shows us that when we introduce GBA into a specific tissue that is distant from the brain, there's actually a rescue of aggregation in the brain. And there's a very dramatic decrease, as you can see in these whole brain images. And also, we find that this rescue is mediated by alterations in the vesicular trafficking pathway, and specifically by alterations in extracellular vesicles. So this leads us into our current model and hypothesis, which is that extracellular vesicles are a vehicle for propagating protein aggregation from cell to cell, and that GBA accelerates this process. And we wanted to study the vesicular trafficking important for lysosomal degradation and extracellular vesicle formation to see if those pathways were altered by GBA. So the first model we use in our lab is actually the Drosophila fruit fly GBA loss of function model. So in these flies, we delete the Drosophila homologue of human GBA, which is 50% similar and is expressed ubiquitously. And in these flies, we express human alpha-synuclein, the protein I said was in Lewy bodies, in the muscle and actually the neuronal tissues. And we age them a specific amount of time. It's sort of a long process. And then we separate the neuronal and muscle tissue and run them on a Western blot. We use actin as our control and synuclein as our marker of interest. Synuclein is a very small protein. So GBA deletion causes these proteins to aggregate. And so we see when there is aggregation, these higher molecular weight synuclein oligomers, as we'll see on the gel. And we look for also the low molecular weight synuclein, which is indicative of when there are the synuclein monomers. So yeah, this is the Western I ran actually just last week. On the left, we have the heads, and on the right, we have the thoraces. Unfortunately for this gel, the actin control bands did not actually show up very well. So it was difficult for us to normalize the signal we received from the synuclein. We also saw much less of the high molecular weight synuclein than we expected, especially in the GBA deficient flies, and much more of the low molecular weight synuclein. So future steps for this project would just be to rerun the gel, hopefully with better control lanes, and just to be able to see whether there actually is a difference. The second model we use in our lab is the human neuronal GBA Parkinson's model. For this model, we collected fibroblasts from a Parkinson's patient carrying a mutation in GBA, and we produced induced pluripotent stem cells, or iPSCs, from them. We also used those same cells to produce wild-type revertant cells as a control, and collected from a different individual age and sex-matched healthy control cells. So what we did with these cells is we differentiated them into neurons, also a very long and complicated process. We fixed and stained them with different lysosomal markers, each corresponding to a different stage of the vesicular trafficking pathway, and we imaged them using confocal microscopy. So the cool thing about confocal microscopy is that you can focus light through a pinhole to focus on a specific Z plane. So these are what are known as Z stacks, which are a bunch of those planes stacked on top of each other. For these specific cells, on the left we have wild type, on the right we have mutant, and they are marked in green with the lysosomal marker EEA1, which marks the early endosomes. And for each of the 5 lysosomal markers, we imaged 15 neurons per cell line for a total of 150. So these are just 2 of the images I took from, all of those cells. And I think it's really interesting that you can see on the wild type cell, there seems to be a stronger green signal. And I think that is sort of the overall trend I noticed while I was taking those images. But the next step for this project would be to do a normalized quantification of the relative signal strengths in order to be able to make a more definitive statement. All right, I just would like to take this time to acknowledge everyone that made this program possible. Thank you to Dr. Ellenbogen, Dr. McDonald, Dr. Davis, my PI, and Sylvia and Julie for everything you've done. This was a really great summer, so thank you.
And thank you to the Kwan family for coming here. All right, our next speaker is Eric Lassiter. Eric is also a rising. Did you know each other before? No, isn't that interesting? To University of Washington, a rising senior, and he worked in Manny Ferrera's laboratory, and the Ferrera lab is here. So come on up, Eric.
Hello, everyone. Thank you all for coming. My name is Eric Lassiter, and yes, I have been blessed to spend the last 8 weeks in Dr. Ferrera's lab, where I've been able to learn about pituitary adenomas and them specifically at the single cell level. So a little bit of background on pituitary adenomas is that they are these benign tumors arising from any of the 6 major cell types of the pituitary. And despite being benign, they can present with these severe comorbidities due to this hormone oversecretion from the tumor. And one of the biggest things that I want to highlight is that within the literature, they are thought to be monoclonal in origin. However, this can raise some questions when we start to look at the treatment of these tumors, and specifically at the current chemotherapy agents, which are currently only able to sort of suppress this hormone oversecretion, but are not really able to promote any cure or prevent recurrence. And it's because of this lack of efficacy that they tend to be sort of the last resort of treatment, secondary to surgery and radiotherapy. However, once surgery and radiotherapy fail, patients are often found in this sort of battle between the symptoms of the tumor hormone oversecretion or potential side effects and the cost of prolonged chemotherapy usage. So within Dr. Ferrer's lab, in this pursuit to identify better molecular signatures of these tumors in order to promote more targeted novel therapies, they came across this hypothesis that these tumors aren't actually monoclonal as they are conventionally prescribed. And if they were able to find a heterogeneity of tumor gene expression, it may explain the lack of efficacy for the current chemotherapeutics that are available and help us to guide help guide us to have better treatment approaches in the future. So in order to identify heterogeneity within a single tumor, the lab employed this methodology. It's called single nuclei RNA sequencing, which works to profile the gene expression of single cells by isolating the nuclei from these cells and then sequencing the mRNA that is found within. It is extremely advantageous, obviously, because you are able to compare the cell-to-cell transcriptomics within a single tissue. And if you find these differences, then you know that it would not be monoclonal. So the portion of this procedure that's done within the lab entails sort of the sample preparation. Isolated nuclei from frozen tumor tissues are partitioned into these little oil droplets that will also contain a single bead, a single gel bead. And these gel beads contain the necessary probes in order to bind to the mRNA, complete reverse transcription, and then also add these barcodes and adapters and indices for sequencing and then also downstream nuclei tracking. So once the complementary DNA has been synthesized and then amplified to an adequate concentration, it is then sent off to Northwest Genomics for the sequencing portion. And so once the sample libraries are introduced into the flow cell in that top left photo, the forward and reverse strands of the complementary DNA template are synthesized and then amplified via bridge amplification to generate these clusters. And then the actual sequencing portion occurs in these cycles where these fluorescent these nucleotides with fluorescent dye tags are introduced. And as they bind in complement to the strand, they emit this light, which has a very characteristic wavelength and intensity that can be recorded. And then after all of the reads have been sequenced and the recordings are converted into their respective nucleotide bases, we here at the lab will receive all of the reads and then process them through various statistical analysis pipelines, which work to properly align the reads, filter out the poor quality data, be able to scale it, perform this linear and nonlinear dimensional reductions, complete biomarker testing on the clusters, and then also do some cell type annotation. So these figures here that I'm showing you are from an actual pituitary tumor tissue that was sequenced. In the top left, we are looking at the before and after of the data within the quality control step of the pipeline. In the top right, we are looking at the linear and then nonlinear dimensional reduction step of the pipeline, which you can already see and appreciate sort of the different clusters within this tissue that are showing a difference in gene expression. And then on the bottom portions here, in the center is the cell type annotation portion, which is identifying the specific molecular signatures of these different cells within the one tissue that are different. And on the bottom right here, we're looking at a series of genes that are upregulated in some clusters while not in others. And so what all this shows us is that it provides evidence against, or it provides evidence to show that not all pituitary tumors are monoclonal in origin, and also provides a series of potential gene targets for therapies in the future. And then I want to thank so many of you for such an unparalleled opportunity and experience that you've blessed me with this summer. Dr. and Ms. Elbogen, thank you so much for having the vision and compassion to start this program many years ago. Thank you to Dr. McDonald, Jim Pridgeon, the entire selection committee for seeing the passion of neuroscience within me. Thank you to Dr. Ferreira, Dr. Parada, Dr. Duvari, and the rest of Dr. Ferreira's lab for being such generous hosts and mentors to me. Thank you to Sylvia and Julie for all of the hard work that you have placed into creating such a meaningful experience for us students. And thank you to the entire neurological surgery department, the donors of this program, and the rest of my 2022 cohort. Thank you.
Great job, man.
Thank you.
Okay. Couple more.
Thank you. I appreciate it.
Great. All right, our next, Shakima. Yep, come on in. And she's going to Oakwood University in Huntsville, Alabama, and she also is a rising senior.
Okay. Oh, my math. Sorry. 1 second. Okay. Good afternoon, everyone. Before I start, I wanted to give like a special, you guys, Caroline and Eric, y'all did amazing. The rest of our cohort, you guys are going to do amazing too. Today, I'll be presenting a case study from Dr. Ferrera's lab from DAJ. So I would like everyone to meet DAJ. DAJ is a patient of Dr. Ferrera's who has been diagnosed with a pituitary adenoma, a GH secreting one, as well as an insulinoma. Oh, sorry. Okay. So his tumors reflect the symptomology of MEN1, multiple endocrine neoplasia type 1, and that is a genetic condition that is associated with endocrine tumors. So DAJ has a pituitary adenoma and an insulinoma, but when he was screened for that MEN1 mutation, it was not found in his germline, which means he does not have it. It can only be genetically passed down. So because of that, the Ferrera lab questions if the DAJ has mosaicism. It's not moving forward. Okay, yeah. No, that should be good. So sorry. Yeah, so the Ferrera lab hypothesizes that DAJ has mosaicism instead of MEN1 because mosaicism creates differentiated cells around the body that can create their own structures. So to find this, to test the hypothesis, the Ferrera lab does a process called whole exome sequencing via Northwest Genome. And before sequencing, the samples go through different steps to get the final result of a library, an exome library, and then the exome library is sequenced afterwards. After the experiment, we move on to data analysis. And I know this looks very complicated, but we're going to break it down. Yeah, so first, the data goes through a quality check before pre-processing. And after pre-processing, the reads are aligned to a reference genome and converted to a BAM file, which stands for binary alignment map. Those files then go through another quality check step called base score quality recalibration. And after recalibration, variant calling takes place to find the somatic mutations in DAJ's tumor, excluding the germline because we know that it wasn't genetically inherited. And the variants are then annotated using public databases before they are filtered and prioritized, which will give us the list of the most damaging mutations that are in the two tumors, the adenoma and insulinoma. Okay, so for results, this is an example of how we view the data. So on the leftmost column are the total list of mutations found in both tumors. The next column next to that is the mutations that were found in common between both the insulinoma and the adenoma. And then you see that on the right side, the most right is the adenoma and the middle is the insulinoma. So we found, we looked through this data and we categorize all the different variants as shown there. And we found that there were 4 common mutations between both the insulinoma and the adenoma being TDJ NCOR2, LTBP3, and MBFP12. So to break it down a little bit further, we found that 66 of the variants were found in the insulinoma, 30 of them were found in the pituitary adenoma, and then the 4 mutations were the remaining out of the total 100. And they had a common average allele frequency of about 3%. Oh, the slide was skipped. It was the, yeah. I'm not too, okay, I guess I can tell you, there's no slide for it, there was, I'm so sorry. But because of the low allele frequencies of the mutations, we confirmed that DHA is a mosaic because the allele frequency in these 4 common mutations were 3% versus the other mutations that were found in the insulinoma and adenoma, the allele frequencies were around 20%. So because of that big range of difference, we basically think that he's a mosaic, but we can't confirm that until we do further validation sequencing through bench work, comparing these tumors to other of the same tumors in different patients. And then when we did a string analysis, it's basically when you compare, you put genes into a database and it shows how they are connected. So we put the 4 mutations there and we found no direct mutation, no direct connection, but there was an indirect connection via a different gene, SUMO1. It stands for small ubiquitin-like modifier 1. And there's a connection between TDJ and NCOR2. And we think that because of that, ubiquitination might be a factor and how the tumors are kind of, and the mosaicism specifically in DAJ is kind of disrupting how he's able to live in the symptomology. So yeah, sorry for the hiccup, but I'd like to thank everyone who made this program possible and successful. I've learned so much and I really enjoyed being here this summer. And that's all due to everyone, their dedication, their kindness. Like, I've really enjoyed myself. And I'd like to thank the audience for listening and for your time. Thank you.
And Dr. Ferrer was just texting me, he's proud of both of you. One more.
Thank you.
That's great. Lyndon Williamson is from Texas Lutheran University, and that's in Texas, of course. And she's a rising senior.
Good afternoon, everyone. Today I'm going to share with you the research I completed this summer in the Hofstetter and Kine labs. In particular, I was looking at the effects of glibenclamide on tissue sparing after cervical spinal cord injury in a rodent model. So to start at the very beginning, what is spinal cord injury? There we go. So spinal cord injury occurs whenever there's a trauma to the spinal cord that results in paralysis, altered or lost sensory function, or altered or lost bowel and bladder function. Cervical spinal cord injuries in particular occur in the neck, and they're the most severe type, with people with cervical spinal cord injuries often having issues moving their hands and doing really simple tasks, from feeding oneself, picking up a cup of coffee even, can be really difficult for people with cervical spinal cord injuries. As you can imagine, this is a pretty severe impact to somebody's life. And so we're looking at different sorts of neuroprotection therapies that will help people regain some sense of function after their spinal cord injuries. To do this, we looked at the effect of a novel neuroprotective agent called glibenclamide on sensory, motor, and tissue sparing outcomes following spinal cord injury. So after a spinal cord injury occurs, there is something called secondary injury, which Surami will explain more about in her presentation. But the gist of it is the brain spinal cord blood barrier is broken, which allows for blood products to infiltrate the spinal cord. These are very toxic, and they activate immune cells that further damage healthy tissue. So the SIR1 protein channel is highly expressed in these inflammatory cells, and glibenclamide offers an FDA-approved method of reducing these sorts of inflammatory cells by preventing the SIR1 protein channel from being expressed. So we looked at glibenclamide in particular to help improve functional outcomes in cervical spinal cord injury. Of course, we didn't do this with humans because that would be totally unethical. Instead, we used Long Evans rats, and we had 3 groups. The first group was laminectomy only, second is SCI only, and the third is SCI plus glibenclamide. All 3 groups unfortunately suffered laminectomy at the C5 vertebral level. However, only the SCI and SCI plus glibenclamide groups actually had a spinal cord injury. This was considered a moderate injury. It was about 200 kilodynes of force. All of the rats survived for 8 weeks, at which point they were euthanized, and we analyzed their spinal cords, looking at a couple of key things: the amount of spared tissue, the size of the lesion that is a spinal cord injury, as well as their motor function after the injury. So this is our first big finding, right? So looking at this graph here, R means rostral or towards the nose. This is the region above the injury site. 0 indicates the injury epicenter or the most severe aspect of the spinal cord injury, and C indicates below the injury. Glibenclamide is shown in blue, and as you can see, glibenclamide results in a lot more ventral motor neurons. Motor neurons are important because they help convey motor signals from your brain to the rest of your body. The amount of motor neurons correlate with the amount of motor function that a person has after a spinal cord injury. So we want to preserve all that we can. Glibenclamide had on average 16% more ventral motor neurons when compared to the spinal cord injury only group, which offers potential in humans to help raise the threshold of any sorts of motor functions that they can recover after a spinal cord injury. The second finding is that glibenclamide significantly reduces the lesion size at the injury epicenter, or the most severe part of the injury. This is super important, and as you can see, visually, they're very different, right? The amount of gray matter, which is made up of cells that help support the neurons, is vital to restoring vasculature after spinal cord injury. And basically, any tissue that can be preserved, we want to preserve it. This graph is just showing you what I just said, right? So again, R is above the injury site and C is below. 0 indicates the injury site. And as you can tell, glibenclamide reduced the amount of lesion and significantly spared tissue. On average, glibenclamide had about 33% more spared tissue across the whole 7 millimeters of spinal cord that we analyzed. We did find this to be statistically significant, and this tells us that glibenclamide is really effective in sparing both white and gray matter tissue, not only at the injury epicenter, but further out away from the injury site, which is where secondary injury tends to kill tissue. Now, you might be wondering, okay, I'm glad that these rats, maybe were a little bit better than the rats with only spinal cord injuries, but why does this matter? Well, as I mentioned at the beginning of this presentation, cervical spinal cord injury is debilitating for humans, not only rats. And using rats as an animal model for humans allows increased translatability of our findings to humans. In fact, currently there is a stage 2 clinical trial going on looking at glibenclamide as a neuroprotective agent. Our findings are able to help support the use of glibenclamide as a neuroprotective agent as it relates to increasing the amount of motor neurons, decreasing lesion size, and most importantly, improving motor outcomes after cervical spinal cord injury. This final graph is showing you just how much of a difference glibenclamide makes in a rat's motor function after their injury. As you can tell, glibenclamide, the group in blue, had a statistically significant impact on motor functions, which is ultimately the most important thing for human patients. Our research shows that glibenclamide is effective in rats, and we hope that our findings will help bolster the use of glibenclamide to help people with cervical spinal cord injury be more independent, feed themselves, dress themselves, and have more motor function after their injury. I'd love to thank everyone who made this program possible, in particular, Dr. and Mrs. Ellenbogen for realizing their dream and allowing students like us to have a life-changing experience. I'd also love to thank everyone in the Hofstadter and Kine lab for making this summer a dream for me. And it wouldn't have been the same without their support and companionship over the last 8 weeks. Thank you all so much.
Thank you. You did a great job. All right. Shrami Patel is at Kenyon College in Ohio, and she is a rising junior, right?
Hi, everyone. I'm Shrami Patel, and this summer I had also the wonderful opportunity to work in the Kine Hofstadter lab. And my project focused on, before we can even kind of do neuroprotection, we think about how can we actually characterize that injury. So I was focusing on the significance of gray matter loss on functional recovery acutely after cervical spinal cord injury. So there are about over 300,000 people in the US currently living with a spinal cord injury, and 61% of those actually affect your cervical spine. So it's very important for us to kind of understand the injury before we can even start protecting or treating it. So the main way we can kind of think about and kind of characterize spinal cord injury is that it kind of occurs in 2 phases, the initial and secondary phase. This is like a temporal way to describe it. So it's not initial injury doesn't happen and then secondary does. It goes into one. But the initial injury can be characterized by the direct mechanical injury that causes tissue damage. And it starts with the hemorrhaging and the cell death. And then you move about 4 to 5 hours after the injury, you move more into the secondary injury, where there's microglia infiltration, even more inflammatory cell infiltration, and stuff there is less blood flow. So that is a way for us to kind of see that secondary injury is the more preventable one, and it provides a temporal and spatial window for us to kind of look at. And so another way we can kind of think about spinal cord injuries is looking at the differences between gray and white matter. As you can see in this image on the left-hand side, so the image shows microvessel density in gray and white matter tissue. And as you can see on the gray matter side, there are many more. Actually, it's about 4 to 5 times more microvessels in the gray matter than in the white matter. So if there are more microvessels, it's more vascularized, which means there's more bleeding. Therefore, the hemorrhaging is more serious in your gray matter. So, and that kind of progresses into your white matter, and it's known as progressive hemorrhagic necrosis. And so we already all know this. So what we did, which was very unique with this project, is use ultra-fast contrast enhanced ultrasound imaging to actually see this in long Evan rats in real time while they're still alive. And this enables us to see the perfusion parameters and help us identify risk tissue. So with this motivation in mind, we did laminectomies on long Evan rats between C4 and C6 then conducted baseline scans using this contrast enhanced ultrasound imaging. And then we conducted a spinal cord injury using an IH device, and the injury was placed at the midline segment of C5. So once that occurred, we did more scans after post-injury, acutely after, which was about 30 minutes after the injury, and then again at 4 hours, 24 hours, and 72 hours post-injury. And each one was like a different cohort of rats for each experimental time point. So what we found, we've characterized many hemodynamic changes from that, like from all that ultrasound imaging. But something I found particularly interesting was looking at the perfusion deficit. So, and as you can see, perfusion deficit increased as the time point went on. So on the x-axis, you can see the different time points the animals were euthanized. And on the y-axis, you see the deficit volume by fold change, which is just a measurement to show how saturated the blood was in like a transverse section. So as you can see, as you increase the time, there is more deficit, which kind of makes sense because there's your injury, your secondary injury is expanding, so there's less blood flow. And you can really see that in the scans on the left-hand side, whereas at baseline, the red really shows the blood flow that's kind of going all the way through the spinal cord, like transverse section, whereas at 72 hours, blue is bad. And that's where the injury was placed. And you can see like a vast difference between the baseline scan and the 72-hour scan. So this was great, but we wanted to like further support this hypothesis by doing immunohistological analysis. And that's where my main part of the project came in for the summer. So once the imaging was done, all the rats were euthanized with an overdose of anesthesia at their different time points for 24 and 72 hours. Their spinal cords were extracted, they were frozen in OTC, and then sectioned, and then we did many stains on them, but the two I'm going to focus my talk on are NEU-N and GFAP. NEU-N, which helps determine neuronal bodies, and then GFAP, which looks at astrocytes. So once those slides were imaged using the fluorescent microscope, we can see that you can see at 4 hours, the size of the contusion injury is much smaller compared to its 72 hours, like vastly different. And also, so GFAP is the astrocyte stain, and you can see them migrating at 72 hours towards the lesion site, which is something that is seen in previous research. Whereas like on the left-hand side at 4 hours, you can see that everything else seems relatively fine in the butterfly shape of the gray matter. And also when I was looking actually at the slides on the microscope, this longitudinally, the damage was about 40% more expanded at 72 hours compared to at 4 hours, which really, if we go back, you can see there's about a 40% increase from 4 hours and 72 hours on this graph. So the immunohistological data kind of supports the data we've already seen in the ultrasound imaging, which would be great because that means we can do that in patients without killing them to see what's going on in their spinal cord injuries. So further directions with this is I could see in the microscope like the lesion size was bigger, about 40%, but we would like to do further quantitative analysis on that, looking at neuronal counts and lesion volume to kind of like definitively say those statements. And something else that the lab would like to focus on, and like Lyndon's talk was kind of hinting at, which was like we want to apply neuroprotective drugs to the animals and then do ultrasound imaging and seeing if those perfusion parameters are any different. And then something I would just like to mention is that even though my talk focuses on grey matter loss and a lot of the work in the lab does that, we also acknowledge that white matter is important to functional recovery and neuronal, like how the neuronal networks work in a spinal cord injury. And that is something the lab would like to look at in the future. So I would like to acknowledge everybody in the lab, and especially my PI, Dr. Kine, and Jeff for helping me so much this summer with like all my questions at any time. I would also like to acknowledge Dr. Ellenberger and Dr. McDonald, and a huge thank you to Sylvia and Julie for like organizing anything and making us all feel at home here. So thank you so much.
One more. Thank you. All right, thank you. Great job, Stephanie. What did you? Oh, Julia Walker is next. She's in Bellevue College in Washington, and she's a rising senior, and she's working in Eric Collins' laboratory at the Fred Hutch.
I really appreciate everyone coming out today to show their support for our work that we've done this summer. I'm going to tell you a little bit about an ongoing project that we're working on in the Holland lab that looks at using RNA vaccines to treat cancer. This summer, we focused on targeting gene fusion positive cancers. I was lucky enough to be placed in the Holland lab at Fred Hutchinson, and in the Holland lab, we study brain tumors with a focus on glioblastoma and aggressive recurrent meningiomas. Our research focuses on building mouse models of human cancer that can be used for preclinical testing of therapeutics, as well as analyzing the function of the drivers of cancer, like gene fusions and aberrant splice forms that are found in cancers. The foundation of our model is the RCAS-TVA system that allows us to induce in situ tumors, which develop in 1 to 6 months, depending on the model. So most of us are pretty familiar at this point with how RNA vaccines work, thanks to COVID, and ours works pretty much the same way. The idea is that we're going to harness the immune system to attack intracellular antigens. The difference is that instead of attacking a pathogen, we're asking the immune system to target something that comes from within. And the issue here is that cancer is essentially self. If we tell the immune system to attack self, it becomes highly problematic. So the solution is that we need to target a driver of cancer that isn't found in the body outside of cancer. So in our lab, we've identified fusion genes as a target. They're present in all forms of neoplasia. They contribute to tumor formation due to their tendency to produce these excessively active abnormal proteins. They're commonly found as oncogenes, and they're most prevalent in pediatric cancers. And the way that they work is they constitutively activate oncogenic signaling. So you can see on the right here, sort of an illustration of how it forms. You get these rearrangements of chromosomal or somatic chromosomal rearrangement, which causes the gene fusion, which leads to these fused proteins. Okay, so we have just begun this really incredible discovery study that's going to address the question, can we show immunoprophylactic effects or treatment effects vaccinating against these drivers of cancer? So our goal here is to get meaningful reduction or prevention of tumor growth. So our hypothesis for this very first stage is that RNA vaccines can prevent tumor growth by targeting these oncogenic gene fusions. We've chosen these specific fusions that are listed up here because we work with them all the time, and we know they're very reliable, and we know they're aggressive. So it's going to lead to a really good readout. If this system that we're developing works, this will be the best model in which to prove that. So because this is a discovery study, much of our work this summer has been centered around sort of developing the method and the protocols. This illustrates how we are testing our system. First, we generated this specific gene profile that's going to allow us to run the system effectively. We ran a toxicity study to make sure that the vaccine was safe. We did a tumor growth control study where we induced tumor growth on each side. This is a really critical step because later on, we're going to be showing evidence that's basically nothing. So by doing this, we can show that in the absence of the vaccine, we're still able to grow tumors. And then we're running an assay currently that's going to allow us to show that our vaccination process is effective. Okay, so this is our experimental plan. We have done the first 2 rounds of vaccinations. On Monday, I'm going right back to work, and I'm doing RCAS-TVA flank injections. We want to focus on the flank first because we go straight to the brain. There's an issue of the blood-brain barrier possibly having an effect. So we want to show this works in a flank tumor first and then move on to brain tumors. And then after that, we're going to do the 3rd round of vaccinations, and then we're going to be evaluating the immune response. Okay, so what we're going to see, if it doesn't work, we're not going to see any difference between our control and vaccinated mouse. We're going to go back and reevaluate the design if that's the case. It could also possibly work initially, but then get a rebound in the tumor growth. At that point, we need to start looking at escape mechanisms. If it does work, ideally, we're going to see nothing. The tumor's not going to form, and then we'll be able to do tests and see if we can show an immune response and an impact on the tumor growth. And at that point, we then get to pivot and get a mouse that already has an established tumor and see if we can vaccinate away the tumor. This is really the most exciting step for me because this is how we would treat humans. This is how we treat an actual patient. We're not going to pre-vaccinate people against these tumors. We need to be able to treat a patient. So when we finish our work with gene fusions, we're going to move on to our next target, which is this T1 isoform. It's a really exciting target because this isoform, unlike the gene fusions which are found in these really rare pediatric cancers, we believe that T1 is a driver of many, if not all, cancers. So right now we have a manufacturer working on the vaccine. When it arrives, we're going to be able to hit the ground running because we've already done all of this work through the gene fusions. I would like to thank all of the many wonderful people that have played a role in my summer of discovery. All of my many mentors at the Hutch, including my PI, Dr. Eric Holland, the incredible scientists in my lab, Frank, Debbie, Dennis, Zach, Maddie, and James, and everybody else. This has been a really incredible experience for me. I told Dr. Holland the first time I met him that I felt called to the work. And after this summer, I have realized that this is what brings me purpose. And for that, I am truly grateful to everybody in this room, everybody on Zoom. Thank you so much. I want to thank Drs. Ellenbogen and McDonald, Jim Pridgeon, Julie, and Sylvia, the selection committee, and all of our amazing donors. I'd also like to thank my incredible cohort of students. To quote a very wise woman that I know, this summer with all of you has been a 12 out of 10. Yeah. And then I have one last thank you to my dad. His experience with neurological surgery taught me that you can face this with a sense of humor and curiosity rather than fear. And thank you so much.
For the folks in the audience, that's Dr. McDonald in the autopsy lab. And she took the students to an autopsy lab, and that's a pretty amazing experience. The other side of surgery. So thank you, Dr. McDonald. Couple more, thank you. One more, thank you. You're all doing an amazing job. Carolyn Sutliff, who is in Bowdoin College in Maine. Okay, we have trivia about Bowdoin College. She's a rising junior and she's in the Judavit lab.
Hello, everybody. My name is Carolyn Sutliff, and I had the opportunity to study with the Judavit lab about how the proximity to beta amyloid plaques in Alzheimer's patients disrupts the endolysosomal function of microglia. Okay. So first off, Alzheimer's disease is a neurodegenerative condition which is characterized by tau tangles in beta amyloid plaques. So beta amyloid accumulates naturally over time, just as we age. But with Alzheimer's patients, we see that they are packaged into little plaques. And that's kind of what we see in the second image. And we don't really know the process of how Alzheimer's disease progresses, but we do have one part that we think is a pretty big player in the process of neurodegeneration, which is neuroinflammation. And microglia have been implicated in these because they are immune cells in the central nervous system. So what they do is they phagocytose things that shouldn't be there, like foreign bodies, or they can just clean up the brain. So they protect your brain, keep it clean. They're great. They're very cute too. So they have certain morphologies. So right here, we have the resting state. So that's when they're ramified and they have long processes whenever they're surveying their surroundings, looking for things that need to be cleaned up. But then as we go towards the activation phase, they become amoeboid, and that's whenever they are typically phagocytosing things. So then back here in the circles, those are the plaques in pink, and then the microglia are in green, and they swarm plaques, and they can go inside the plaques or just hang around outside, but they have an amoeboid appearance around plaques. And so, okay, hold on. There we go. Don't know who that was. So this is the endolysosomal function. So this is what microglia use to phagocytose things. So what they do is they can just kind of like take it into the lysosome and then break it down from there. And this is one of the first pathologies that we see for Alzheimer's patients. So this is before we see any beta amyloid plaques or any tau tangles. This is the first part. So the question is it amyloid beta or beta amyloid plaques that cause the inflammation of microglia? So what we did is we got a few, we had 7 cases. 2 did not have Alzheimer's disease, and then 5 did. We had a matched case. So we had basically an equal or almost equal age for the control as in the Alzheimer's patients. So we use immunohistochemical staining and spitting disc microscopy to investigate what's going on with the microglia. So what we do with immunohistochemical staining is that we have an antibody, a primary antibody to attach, and then a secondary antibody with a fluorescent protein. And so here are little proteins. We have microglia in green, which are stained with IBA1, lysosomes in red, which are stained with LAMP1, and plaques with 6E10 in pink, and then the nucleus with DAPI in blue. So in our controls, the microglia are in an inactivated state. So they're the ramified, calm ones. They're just surveying their surroundings. There's basically nothing going on. And if you notice, we have basically no red signals. So no LAMP1. That means that these microglia are not inflamed. And in comparison to, oops, okay, so they're the microglia. So we have these plaques, and they take on a very different morphology in the immediate area around plaques. They become more amoeboid, and you'll notice some inflammation going on compared to the surrounding microglia, which are more ramified and not inflamed. So next, here we have the Alzheimer's microglia. So with these close-up pictures, you can see the LAMP1 signals better, and we see that there is a very big increase of LAMP1 signaling, which means that we have more lysosomes, or we don't really know if they're more lysosomes or if they're just bigger lysosomes. So what they do is they've really swarmed around the plaques, and then they become inflamed with a greater LAMP1 signal. And so this kind of fits along with our theory that beta amyloid plaques cause the disruption of the endolysosomal function, because we only see an increase of LAMP1 in the immediate area around the plaque. So we don't really know why the LAMP1 signal increases. We don't know if it's triggered specifically by the beta amyloid, like it just makes the microglia mad, or if the microglia recognizes it as a threat and overproduces lysosomes to dissolve it. So again, this kind of fits into the whole theory that the beta amyloid is what causes the microglia to become inflamed and cause the neuroinflammation, which then furthers the progression of Alzheimer's disease with neurodegeneration. So some future directions that we can take would basically be to have additional cases. That way we can get better statistics from our group. Another part would be additional imaging. So I believe somebody mentioned having Z-stacks. I think it was Caroline. It would be able to help us figure out if the lysosomes are bigger or if they're just more lysosomes. And then let's see, quantification. So just do some stereology and analysis of specifically the area around the microglia or around the plaques as you get further and further away. So lastly, I'd like to acknowledge everyone. It's been an amazing program. So thank you, Sylvia and Julie. And thank you to Dr. Ellenbogen, Mrs. Ellenbogen, and let's see, let's go with the GIDEV lab next. So over there we have Dr. Prater, who's been amazing. She's my PI, and Lexi. She's been amazing as well. They've really made the summer amazing. And thank you to Mr. Pridgeon, I believe. Okay, and the donors. That's also very important. So thank you to the donors for allowing us to study their the tissue of their relatives. So yeah, thank you all.
Hitham is from the University of Washington in the Columbia Laboratory at Seattle Children's Research Institute. And Frank has hosted a student for the last decade or so. So come on up.
So hi everyone, my name is Haitham, and I was honored to have the amazing experience to work in the Kalume lab this summer. I am looking at the impact of the NDUFS4 knockout on interneural morphology and seizure susceptibility in a mouse model of Leigh syndrome. Sorry. What is NDUFS4? NDUFS4 is a gene that codes for a protein of complex 1 of the mitochondrial respiratory chain. Mutations causing a loss of function in the NDUFS4 gene are linked to Leigh syndrome. Leigh syndrome is a progressive neurodegenerative disorder that is characterized by progressive loss of mental and movement abilities, respiratory distress, and epilepsy, ultimately leading to premature death between 2 to 3 years. Pathologically, it presents with symmetrical bilateral lesions in various brain regions, such as the basal ganglia and the brainstem. The MRI to the right shows a child that has lesions in the basal ganglia. Leigh syndrome is the most common defined clinical pediatric presentation of mitochondrial disease. Knockouts of NDUFS4 is a good model of Leigh syndrome in mice. Mice with the mutation exhibit seizures and other phenotypes of Leigh syndrome. Previous work in the Kalume lab has shown that seizures and epilepsy arise from the impact of the NDUFS4 knockout on GABAergic neurons. And while the other phenotypes of LS, like respiration problems, come from the excitatory neurons. My project has two aims to further understand the mechanism of epilepsy. The first aim is focused on assessing the impact of the NDUFS4 knockout on interneuron morphology and seizure susceptibility and the development of seizures. The second aim of my project is to explore seizure susceptibility when the NDUFS4 knockout is restricted to interneurons in the brainstem and cerebellum. I'm going to go over aim 1 first. The mouse generated for aim 1. The mutant mouse was generated by crossing a double flox NDUFS4 that is DLX1-2 cre positive with a double flox end-of-spore mouse that is homozygous for AI14. Generating a mice that is double flox for end-of-spore, DLX12 Cre positive, and heterozygous for AI14. The controls were also generated in a similar fashion, only they are wild type for end-of-spore. The end-of-spore knockout was generated by Cre recombinase, the protein of the Cre gene, like symboled by the two Pac-Man symbols on the screen. They cut out DNA between two LOXP sites, which it does for end-of-spore knocking out the gene, and the stop codons for AI14, allowing for TD tomato, the fluorescent protein, to fluoresce bright red. So the strategic approach for AIM-1 was that, well, we use DLX-Cre because it targets dispersed GABAergic interneurons in the brain, which allows for better analysis of distinct cell morphology, in contrast to GAD-Cre, which was previously used in the lab. Only it targets all the interneurons in the brain, resulting in a densely packed image, making it difficult to distinguish a single neuron. The protocol used was perfusing the brains and fixing them, collecting brain slices, and using the Olympus size scanner to collect fluorescent images. The images were supposed to be analyzed using Scholl analysis, but we ran into some technical issues and did not have enough time before this presentation. One method of Scholl analysis is measuring the number of neurite intersections from the center of the cell, which is the soma, and counting the number of intersections with, yeah. Sorry. Here are the results from the hippocampus. It shows the cell labeling are spread out as expected with the DLX12 Cre in both the control and mutant mice. You can also visualize some of the dendrites on the cells as well. Now looking at the cortex, you can also see a similar thing as well with the scattered labeling of the GABAergic interneurons. This project is still ongoing in the Kalume lab. Future endeavors will include improving the resolution and taking Z-stacked images for Scholl analysis. Now I'm going to move on to AIM-2 of my project, which is exploring seizure susceptibility when end-of-spore is restricted to interneurons in the brainstem and cerebellum. So the mice used for this AIM was a double flox end-of-spore that is glycine Cre positive, close to a double flox end-of-spore mouse, generating a mutant that is the double flox end-of-spore and DLX Cre positive, and the control is double floxed end-ofs and DLX Cre negative. And glycine Cre was used because it targets interneurons in the cerebellum and the brainstem. The method for this, for testing seizure susceptibility in the mutant mice and the control mice, was done by using a heat lamp and a temperature probe implanted in the mice. We slowly raised the temperature by 0.5 degrees Celsius until a generalized tonic-clonic seizure occurred, at which point we recorded the mouse's temperature and cooled the mouse off. The main takeaways from this experiment is that the controls did not have an myoclonic seizure, which is essentially not a full-on seizure, except like a but an abnormal twitch. 80% of the mutants had a myoclonic seizure, while 20% did not. And at baseline, 20% of the mutants already had a myoclonic seizure. And as we slowly increased the temperature, 80% had a myoclonic seizure. For the generalized tonic-clonic seizure, none of the controls had a GTC seizure, and all of the mutants eventually had a GTC episode as we slowly increase the temperature. The average baseline for the mutant mice was around 37.5 degrees Celsius. The myoclonic onset was around 38.2 degrees Celsius, and the GTC onset was around 40.8 degrees Celsius. In conclusion, for the morphological study, our approach shows that we can label dispersed populations of GABAergic neurons across multiple brain regions. The neural dendrites are visible in our images and control and mutant mice. However, for optimizations are needed to improve the image quality for optimum soil analysis, and acquisition of Z-stacked images will improve the resolution of neurites. For the seizure study, a mice with the end-of-4 in the brainstem, GABAergic neurons using a glycine Curie driver developed high susceptibility to thermomyoclonic and GTC seizures. This indicates that GABAergic neurons in the brainstem play a key role in the mechanism of Leigh syndrome related epilepsy. So I would like to give a huge thank you for everyone who supported me throughout this program, and specifically Dr. Ellen Bogan and Mrs. Ellen Bogan, Dr. McDonald, Jim Pridgeon, everyone in the Kalume lab, Dr. Kalume, Michelle, Arena, Judy, Natalie, and everyone in the Department of Neurological Surgery, and of course, Sylvia and Julie. Thank you.
Very nice. Some more. Got it. Thank you.
Thank you.
All right. We're going to go on Zoom for the next one. Franklin is in Denison, and he is a counselor, so he had to be back to welcome the freshmen back. So ready to go, Julie? Okay. Dre, are you ready?
Yes, I am.
Beautiful. I feel like you're talking from above. Oh my gosh, go ahead. Go, Dre.
Okay, I will. Yeah.
Well, is Dre sharing?
Can everyone see what I'm sharing?
No, not yet. You've got to share.
Okay, I will stop for a second.
We see his slides.
Okay.
Her slides are done.
On Zoom, we can see his slides. Okay, but we can't see him here.
On Zoom, you see the slides.
Yeah,
Yes.
we see your slides in Zoom. Are we on Zoom here?
This is meeting.
I think we'll have to close the meeting.
Oh, I see what you're doing. Is there what? No, nothing. Okay, so. There we go.
Yep,
Okay.
there we go.
There we go.
It's back there somewhere. Okay, go ahead, Jay.
Okay, thank you. Hey everyone, my name is Franklin Bray, and I worked in the Ed Lean lab at the Allen Institute for Brain Science. For the last 7 weeks, I had to leave a week early, as Dr. Ellen Bogan explained. I want to begin my presentation by talking about single nucleus RNA sequencing. So this is the technique that basically allows us to see the gene expression levels in certain cell types, in very specific cell types. Hodge et al. Is a paper that shows us that we don't actually need the entire cell to understand what's going on and to subtype cells, we only actually use the RNA. So this entire process comes up by getting a brain sample, homogenizing it, which means breaking it down to small bits, going through a few gradient steps where you separate out the different areas that you want, and putting it through a fax machine, which means that you stain your sample first, put it through a fax machine, and then you get, after analysis, these plots that you can see at the bottom of the screen. What I want to bring to your attention is this small spot here. We can see excitatory, which is this huge population here, a smaller population inhibitory, and this small, very small population, which makes up about 1% or less than 1% in most UMAPs, which is a non-neuronal population. So how do we enrich the population of non-neuronal cells? This is where VineSeq comes in, which is vessel isolation and nuclei extraction for sequencing. So a general introduction to VineSeq. It was established by Dr. Andrew Yang, who is a professor at a medical school, Stanford Medical School. And his technique, the goal of his technique was to increase the representation of the nuclei from vascular cells for RNA sequencing from postmortem brain tissue. His paper showed, once he had used this technique, that 30 to 45 of the genes, the top genes that are linked to Alzheimer's, could be found in the human brain vasculature and could contribute to your AD risk. So that said, my goal at the Allen Institute was to establish this method in their lab so that they could do similar studies and run similar analysis. I also wanted to bring your attention to this figure from his paper. This green population, so we're moving from this small population that you saw in the first figure to this, where it's like 36.8% of the UMAP representing non-neuronal and specifically vascular cells, or nuclei from vascular cells. So the first stage in this entire process is called vessel isolation. First, like I said, for normal RNA sequencing, you take your brain sample, you homogenize it. For this experiment, we'll use a dextrin gradient, which allowed us to get a pellet, this small pellet at the bottom, which is what we want. We go through centrifugation twice to get what we want. And the product is kind of this. You get a sample, you dye it, and you look at it under a microscope, and this is what you would see. This is the vasculature that is normally lost in normal nuclei isolation techniques. But what I also want to point out is these small dots in the background, which are nuclei that we don't really want from neurons, from other cells in the brain. We want to remove that. So what we use is a filtering step. And in this filtering step, we apply the sample, which has been downsed and gone through the spinning step. We resuspend the pellet and we put it on a filter. And then we wash that filter with PBS, with a blocking buffer, to basically wash away all those other nuclei that we don't really want. And to keep, the idea is that the vessels would remain on top of the filter and all the other nuclei that we want would wash out into this bottom fluid that you see here. Once we have this filter and the vasculature is on top of the filter, we need to get the vasculature from that filter. And we do that using a shaker method, which my lab and I kind of created on the fly. Basically, you put the filter in a vacuum tube and you shake it for 30 minutes. I'm doing it manually here because the shaker tray that we normally use was being used by another lab at the time, another researcher at the time. But basically, I spun that for 30 minutes manually. And then the result is this, where we have a much more concentrated population of vasculature. And we also have all those background nuclei and so that we don't really want in our analysis completely gone. When we finally got this photograph, I was jumping around my lab because this was about the product of like 4 weeks of troubleshooting and everything and finding out what was working and what was not working. But everyone was in a meeting, so I was just jumping around alone. So pre-shaker method, this is what we would have, where you would still have all this background. But basically, this is a picture from the flow through of everything that would fall through. And we would wash so much that we would have the nuclei going through, but we would also have our vasculature breaking up. So instead of staying on the filter where we want it, it would fall through in the small fragments that you can see at the bottom. I think you can see a little bit here. And we don't want that. So the shaker method was really a big improvement and a big step towards getting what we actually wanted. Right. So these are FACS plots. FACS plots are, sorry, okay, right. So FACS plots are fluorescence activated cell sorting. It's a machine that uses light, shoots beams of light at cells, and basically uses that to determine what type of cell they are once you have stained them with special antibodies. So this top population represents neurons, the neuron population. This would be oligodendrocytes, and this would be all the non-neuronal nuclei that we have in our sample. And what you would see is that this is the population that we would normally get when we use Dr. Yang's method. And we were like, what's going on? Why do we have all these neurons? Why do we have all these oligodendrocytes? And what we did with one of our methods, before we actually finalized anything, which we still haven't, is that we were able to greatly reduce the amount of neurons that we got on nuclei from neurons, greatly reduce that oligodendrocyte population, whilst maintaining a decently sized non-neuronal population. So as you can see, the general population of nuclei that we have is reduced, and we're working on maintaining the size of that population. But as we lose neurons and as we lose oligodendrocytes, we are bound to lose overall population numbers. So the future of VineSeq. So now that we're able to isolate vasculature, the Allen Institute is working on nuclei extraction techniques. Because now that we basically have the cells, now we need to work on breaking up those membranes and keeping those nuclei intact, maintaining the integrity of the RNA inside of them so that we can do analysis for Alzheimer's research. An example of a solution that we have thought up is of using syringes to suck up and break up the nuclei. The cells, while maintaining the nuclei, the integrity of the nuclei. We're also looking at using stronger detergents to break apart tissue. And we're also, part of my project was going to be, before we found out how troublesome this whole process would be, protocol would be to use VineSeq to analyze the vasculature of persons with and without Alzheimer's as a way of basically confirming the results of Dr. Yang's paper. I did not know I would have this much time, so I'm going to go back and talk about this for a second. Here in the shaker method, we use detergent in the vacuum bottle to break up, to basically destroy the nuclei in there, which is how we were able to get this clean presentation here. The detergent in here is basically destroying everything that we don't want. And what we basically found is that the vascular cells are basically indestructible. Nothing will destroy them, not stronger detergents, not manual force, using a plunger of any sort, of anything. We've thought about using time. We've given them like 10 minutes, 15 minutes with strong detergent, and they remain basically impervious to any form of attack. So that's my presentation, basically. It's very different from everyone else's because my project was basically working on maintaining, basically establishing a method. This picture shows these little fragments that you see that look like scratches on the bottle are actually broken apart pieces of vasculature that survived detergent, have survived mashing, have survived everything that we have thought to throw at them. So this is where we are right now. And we're working on breaking those apart, getting those nuclei out, maintaining that RNA integrity, and hopefully confirming the results of Dr. Yang's paper. I would like to thank everyone in my lab, Dr. Ed Leigh for having me in his lab, Dr. Rebecca Hodge for being here and for being there for me throughout the weeks, Jeremy, who taught me everything I know about RNA analysis, even though I didn't get to use it because of the timeline of our project, but he was with me every week, and JT, who was in the lab with me almost every single day, helped with my ideas and everything. I also like to thank Nadia, who was there with me for when we did our FACS plots. She explained everything to me about how FACS plots works and everything. Okay. And Al, who greeted me at the door every day. I'd like to thank the NIH and the Nordheim interns for supporting me when I came back basically home every day. And I'm like, my experiment failed again. So thank you guys. I would like to thank Dr. Ellenbogen, Mrs. Ellenbogen, Julie and Sylvia for everything. They were so helpful in everything. Jim Pridgett, Dr. McDonald, who was so motivational every time we saw her. Yeah, thank you. I was under the impression that we would have questions, but we don't. So I didn't want to show this picture, but yeah. Bye, everyone, thanks.
Wonderful. Great job, Franklin. And I have your certificate, so you're gonna have to come back and get it. So, all right. All right, our next speaker is gonna be Abigail and Natalia together. Wow, it's great.
You're not in presenter mode in the Zoom.
Oh, not in presenter mode?
No.
Nino, a voice from above. Oh my God.
Now I scared you.
No, you did,
Oh.
for sure. You did. There he is. We can see you beautifully. How about now?
Perfect.
There we
All right, thank you.
Good luck.
Thank you. Okay. Hello, my name is Natalia,
And I'm Abigail.
and we will be presenting on chronic intermittent hypoxia and how it disturbs the regulation of breathing and drinking in awake and alert. Oh, I have to talk closer. Okay, alert mice. And we were working in the Ramirez lab. Yeah. So what we know is that, hello? Oh, okay, cool. Okay, so what we know is that 25 to 45% of pediatric cases deal with dysphagia, which is a swallowing disorder that affects breathing. And that, oh Lord, okay, what is this? And that 3% of pediatric cases involve obstructive sleep apnea. And of these are comorbid to other disorders. Where is this going? Okay. To other disorders such as congenital heart disease, neuromuscular/neurodegenerative disease, craniofacial abnormalities, and obesity. It is important to note that these are also prevalent in adults. In fact, they're the #1 leading cause of death in neurodegenerative disorders.
Chronic intermittent hypoxia, or CIH, is a widely used model that mimics obstructive sleep apnea. Obstructive sleep apnea, or OSA, is the obstruction of the upper airway during sleep. The closure of the airway occurs during intermittent pharyngeal collapse. And on the left, you can see a diagram where it details where in the pharynx this collapse occurs. To the left of this, the right of the schematic, is an anatomy diagram. And here we want to point out that it shows the upper airway as well as the lower airway. We also want to note that OSA is a human disease. As mentioned, CIH is a model for OSA, and the inspiration for our project actually came from an article that was published by one of our mentors, Dr. Huff. And this article looks at the interaction in swallowing, breathing coordination. What we currently know is that dysphagia, or disordered swallowing, can lead to aspiration pneumonia by not being able to coordinate swallowing and breathing. Here in this diagram, aspiration pneumonia occurs when food or liquids travel through the trachea to the lungs rather than
traveling through the esophagus during normal swallowing. Aspiration pneumonia is also a leading cause in death in breathing and neurological disorders. What we currently don't know is if licking drinking coordination with breathing is disrupted in OSA. And this leads us to our hypothesis that chronic intermittent hypoxia disrupts coordination between licking and breathing.
And then this leads us to our chronic intermittent hypoxia protocol, which we used in 2 chambers. The first being what you see in red on the diagram to the right, where it's cycled between 5 minutes of 21% oxygen and then 60 seconds of 5% oxygen at about 80 times per day for a total of 23 days, starting at 9 in the morning and ending at 5 p.m. in the evening. And then the control chamber just remained at 21% oxygen for all 23 days. To ensure the health and safety of the mice, we did weigh them daily, and their cages were changed weekly. We also used adult mice at P40, meaning postnatal. And then for our experimental timeline, all the purple regions are the time that they spent in the chamber. The green portions are the water deprivation periods. I would like to note that they were water deprived in order to have them get used to licking in terms of the chamber that they were put into. As a reward system, they would get sugar water. However, they did receive a daily set amount of water every single day during that time period. And then in the blue region is the free water they received during the weekend, and the yellow and red are the testing period days. And for those of you curious about the data, we used a total of 15 mice on a black 6 background, and then details on sex distribution are in the table to the bottom left. And then we used, oh, that was a little wonky, but it's okay. We used this setup as our lickometer and to acquire plethysmography. So that blue stuff that you see at the back behind that clear chamber is what records our plethysmography, which is highly sensitive. And where the lick spout is what records when they lick. So that's their lick sensor, and that's traveling towards that green box over there. And I'll show you what that reading looks like in a second so that you understand our data a bit better. And then what we did here was run 2 trials of 3 minutes each. We didn't do 5, even though we originally were going to, because we found that after 3 minutes, they were satiated and no longer wanted to drink. And when they when they stopped, they would either move around, jump, or do other behaviors that, because of its sensitivity, would kind of skew our data. So keep that in mind. And then as a note, the knitted mouse is not to scale. They are much smaller in real life. And this is a plethysmography example. Just for simplicity, we'll use the word pleth. And here it's a very zoomed in version. We'll zoom out a bit more when we go to our data. But just so this is for your sake to make it easier to understand, the x-axis is time and the y-axis is the change in pressure in terms of the sensitivity of the chamber. Upwards motion is inspiration and downwards is expiration. And then something to keep in mind is that this has never been done before. So keep that in mind as we go through our data. And then, as I said, we measured lick and non-lick bout durations. The orange portions in the lick sensor is what we measured. The blue ones are as lick, and then the orange ones are as non-lick. And it corresponds to the readings in pleth as blue and orange. And we use the root mean square amplitude. And you can kind of already see that there is a difference going on. In blue, the lower change in pressure implies that it's shallower and shorter breathing. And just as a reference, we used the percent max of baseline of the non-lick in order to compare our data. However, because of that added noise and external factors, some of the readings could have been skewed. So take that into account.
And here we have a pleth reading from our control group. We want to note that we didn't see a difference in the pleth reading, the breathing in the mice from comparing non-licking to licking. And we noted that this was reflected in our data as well. There's no significant change in this group comparing licking to non-licking, and there's also no difference in the in breathing in these mice and the root mean square amplitude as well. Here we have another pleth chamber reading. This one is from our experimental group, the CIH mice. These mice were hypoxic. And what we noticed with these mice is that there was a difference in the breathing amplitude in these mice when comparing licking to non-licking. It is significantly lower, but it returns to baseline when they are done licking or close to baseline. This is also reflected in our data. These mice, we noted that they spent less time licking than not licking, and there was a significant difference between licking and non-licking in the bout duration. And there's also a significant difference in the root mean square amplitude as well. And this bottom graph here, we decided to look at the lick bout duration between these two groups and found that even though it looks like there might be a difference, there actually is no statistical change between these two. In addition to looking at coordination between breathing and licking, we wanted to look at the sex differences between these groups as well. And we actually noted that there were no differences between these groups, but we also looked at the coefficient of variation and found that males had a higher variance in lick bout duration than the females did.
And then for limitations, like I said, because of how messy it could get because of external factors, we ran an outlier test on our root mean square amplitude calculations to account for all of that that could have disrupted our data. And what that kind of looks like, just to give you a visual image, is what is on the bottom, a messy trace versus a clean trace. And secondly, for trial and error, we actually ran this experiment twice. In the first set, it was our first time, so we were still trying to get used to blocking out as much noise as we could. And then the second time, we perfected it a bit more. So you can see that the traces would have improved, and that could have played a factor in our data. So just letting you guys know about that. And then as for sample size, we used a total of 15 mice, which is not relatively small for mice, but could always be better in terms of acquiring data. And then for interpretation, we found that chronic intermittent hypoxia not only disrupts swallow breathing coordination, but also drinking/licking breathing coordination, which is really fascinating because breathing as a whole field has not been studied as much. So to try to measure that via licking, and then on top of that using chronic intermittent hypoxia, is extremely cool in our opinion, because we geek about this, but also it raises a lot of discussion questions in the Ramirez lab, such as if what really accounts for that female/male data set variability, if there is a specific developmental period in which this apnea effect worsens, and more. So just, yeah, keep that in mind.
And there are a few other things that we actually wanted to share today. There were the things that we learned in the lab and also other projects that we worked on. And one of the things we wanted to share was that we learned how to surgically implant a trach tube into live mice under the guidance of Dr. Huff. This is a schematic that shows where we would place a trach tube, as well as muscles that we record from using electrodes. The second project we worked on was tailored by Dr. Bush. I do want to mention that this was done before, not exactly the same. We looked at if mice liked a high oxygen or normal oxygen environment compared to a low oxygen environment. So you can see that normal oxygen would be 21% and low oxygen would be 10%. And the bottom, you can see this is our data. We're not really going to jump into that given time constraints. The last thing I wanted to share was that we worked on a head fixing project. This was developed by Dr. Bush as well. It is a pilot experiment. He's still working a bit on it, but we wanted to look at recording neurons in the forebrain of mice that could possibly be related to breathing, as this is not something that has been studied or found at all. We invite anybody that is curious about these projects to ask us questions at the end after all the students have presented.
And then these are our references and our acknowledgements. So we'd like to especially thank Dr. Ellenbogen, Mrs. Ellenbogen, Dr. McDonald, Mr. Pridgeon, Julie, and Sylvia, because you guys are awesome. And everyone in the Ramirez lab, 3 of them are here. Hello. We have we have Dr. Huff, Dr. Bush, and Dr. Glover. Yeah, and then there's more listed above. So yeah, thank you so much. Thanks.
Couple more. With two in the photograph, someone will blink. With three, it's a certain. Great job. Great job. All righty. Ariana and Ola. Ariana's at the University of Washington. She's a rising senior. And Ola is a Carnegie Mellon rising senior. And I'm sorry, Abby, she graduated from Wazoo, Washington State University this year. Congratulations. And Natalia's at Santa Clara University, and she's a rising junior. So Ariana, and there you go. Great to see you.
Hi, everyone. Just to reintroduce myself, my name is Ola Raduni, and my name is Ariana McLean. We had the pleasure this summer of working with the SANS lab, and we would like to go over cerebral vascular complications and dural venous sinus segmentation. So one of the first projects that we got to work on was the blood biomarker study, which focuses on stroke. So specifically ischemic stroke, which is the narrowing and or complete blockage of blood vessels, which leads to a lack of blood flow that will ultimately lead to neuronal cell death, which will lead to temporary or permanent disabilities like weakness or paralysis. So stroke is one of the leading causes of death among adults. So it's really important for us to understand the mechanisms behind it and kind of just understand the research. So other research focuses on blood biomarkers of stroke patients, but they compare stroke patients to healthy patients or patients who just haven't had a stroke. What makes our research unique is that we have our own patients be the controls. So we compare their blood biomarkers before they had a stroke and if they were to have a stroke. We could do this because aortic hemiarch replacement surgery was found to increase the likelihood of a patient having a stroke. So we chose to use them as our population. So to do this, we collect blood and urine samples throughout the surgery from the aortic hemiarch replacement patients, but we also collect from patients undergoing coronary artery bypass grafts because they are having heart surgery just without the risk of stroke. So we can compare their blood markers to make sure that what we find might not be attributed to having heart surgery and more so to having stroke. So we detect these blood biomarkers that may indicate stroke, and some of them are shown up here. And we really want to focus on early detection of stroke because it's really important that you catch stroke early and you're able to treat it, because the longer the stroke occurs, the more damage that can be done. And we want to do this through developing a blood test. That's the ultimate goal. But right now we're kind of just looking at the mechanisms behind it. So urine is collected before and after the surgery, and that's immediately stored. And then we isolate plasma and serum from blood in order to, through centrifugation. And then we take those samples that we've prepped and we work with Olink to undergo proteometric analysis. We're looking at 200 proteins of interest currently. And then after the patients undergo the surgery, a couple of days later, they undergo a research MRI. And this is basically just to look for ischemia. And it turns out that a lot of the patients actually show some degree of ischemia. So the overall goal of the study is to really focus on quick detection of stroke so then we can increase patient outcomes and make that more positive with less damage. Another study that we had the chance to work on was the biopsy study. This study focuses on understanding the genetics and microbiology behind cerebral vascular diseases. And we do this through the analysis of biofluids and tissues. The study is broken up into two parts, but the first part I want to talk about is the endovascular biopsy part, which we look at materials from endovascular surgeries. This mainly includes the coils, which you can see over there in the aneurysm. So the coil is used to kind of block blood flow from the aneurysm. This is just to prevent rupture. And we have to make sure the coil fits or else some blood gets in there. So we have to, if the coil doesn't fit, originally, they might take it out. And so we collect that coil and we isolate cells off of the coil, doing antibody staining, endothelial cells. And then we do flow cytometry on the cells, which basically measures the physical and chemical characteristics of the cells. And then we run 10x genomics, which is RNA sequencing, to understand the RNA expression. And the overall goal of this study is to really understand how cells respond to changes in blood flow. And we hope to be able to understand that because we want to, understand the mechanisms and predict when an aneurysm might rupture when there is that change of blood flow.
So some other collections of the tissue for tissue biopsies were from a craniotomy for a cavernous malformation resection of the left anterior temporal lobe and an aneurysm dome from an aneurysm clipping of the left M2. Those collections are placed in a freezer at -80°C, and those go to creating a tissue bank for further cell research into cerebrovascular diseases. Our final study was on the fluid modeling of cerebral blood flow. So how many people know what CFD is? Great. So it's computational fluid dynamics, which is a tool to help us better understand neurological disorders. It's a non-invasive way of measuring velocity, pressure, and fluid shear stress of inside blood vessels. So as you can see in figure 1, I have a depiction of a blood vessel, and on the inner portion of the vessel, it's red, which correlates with a higher velocity. And as you move closer to the wall of the vessel, it turns green, which is correlated with a lower velocity. And this is due to the frictional force that the fluid feels and causes a drag in the fluid that then causes a lower velocity, shown green. And so this CFD details the blood flow to help with the treatment and outcome of patients. As such, in figure 2, you can see a blockage of the left transverse sinus. And right here, I have a beautiful shear stress equation that I know everybody's super eager to hear about. And so a CFD of the dural venous sinuses is a computational fluid dynamic of the dural venous sinuses, which are the sinuses that drain the venous blood from your brain. And you can see where the location of the dural venous sinuses is in figure 3. And below that, I have a CFD of the dural venous sinuses that will be used to calculate the high pressure differentials in cases as such, like idiopathic intracranial hypertension. You have treatments that include a shunt, which is a tube placed in the brain to help drain fluid away, or a stent, which is used to relieve stenosis. So by measuring these high pressure differentials, we can then determine if a patient is eligible for stenting, which is more ideal, versus a shunt. So the objective is to automate CFD to provide clinical utility. And to do this, we need a large data set of manually segmented anatomy to train machine learning that will then go into automating CFD for clinical usages. So this last summer, we used a program called VMTK, and that stands for Vascular Modeling Toolkit, and that manually segments the dural venous sinuses that then go into training, training machine learning, and then that automates CFD for clinical usages. Which basically means we took a magic school bus ride from the superior sagittal sinus to the jugular veins. And with that, our sincerest appreciation to the NSSSP program. We couldn't have done it without you guys. And thank you for your support and teaching us.
It was truly an amazing summer.
Justin is last but not least from Pacific Lutheran University in Washington State, and he's a rising junior, and he worked in the Weinstein lab. And you can tell everybody's having a lot of fun giving these talks, so that's great.
Okay, hi everyone. My name is Justin, And for the past 8 weeks, I was working in Dr. Weinstein's lab under my PI, Dr. Ashley McDonough. And I'll be talking about my preliminary study on microbial activation in a piglet model of gut-brain axis inflammation. And to kind of give you a bit of background on our study, we have to go to the NICU, where it's found that premature infants have a high incidence of cognitive, behavioral, and motor deficits. And this is typically caused due to inflammation, which is also increased due to high susceptibility of infection and underdeveloped intestinal barrier. But it is also found that the diet of a premature infant can have benefits to the actual neurodevelopment, but the mechanism is unknown, which is what we study in this project. And this is also a collaboration with the Chen Lab at the University of Idaho, where we use piglet models. And the reason we use piglet models is because they are the best non-primate model, and comparing it to rats, it is better genomically, gastrointestinally, and the neuroanatomy too. And in that top right corner, you can see that the piglets, there was 2 sets of piglets, one for human milk and one for infant formula. And these piglets were fed over a span of a month or 28 days. And then tragically, they were euthanized. But in the name of science, they were then harvested. And the Chen Lab focused on getting the gut sample and blood samples, in which they studied the inflammation, the systemic inflammation, and how that affects neuroinflammation. And then we at the Dr. Weinstein's lab focused on how the effects of human milk reduces neuroinflammation. For my section of the project, we focus specifically on microglia, which are the neuroimmune cells of the central nervous system, which are a key indicator of neuroinflammation. And as you can see in the top right, that's one of the major indicators right there, where in the surveying section, you can see the highly ramified branch points in the resting area. And then once activated, these branch points retract, and then they turn into an amoeba-like state. They also release cytokines, but our main focus is proliferation, where we believe that infant formula group will have more inflammation, which would cause more proliferation of microglia and more activation overall. And in order to get the actual analysis, we have to begin by cryosectioning, which is taking those brain samples harvested from the Chen Lab, and we cut it up into thin little slices, place them on little slides, and then do immunohistochemical staining, which involves the primary and secondary antibody, where the primary targets the primary antigen, and the secondary is a little fluorescent indicator to find the primary antibody. And what we use for this is IBA1, which stains microglia, Chi-67, which is for proliferative cells, and DAPI, which stains DNA. And in this image, you can see these colors in their respective antibodies. And in order for me to get this actual imaging, we go to the Mariana's microscope, and you can see the microglia in this image are red, which indicates that they are proliferating. And once I have these images, I get something like this, which is called a montage, which is a greater image of the actual microglia. And this is specifically the right prefrontal cortex. And I do something called serology, where I get a little mask of the area, and then I allow the computer to calculate random areas for me to do unbiased quantification. And all these little grids that you see in the top right are those zoomed in on the top left or the bottom left. And they have a little 3D plane in which I can scroll through and quantify all the cells. And with this, I found that after doing a t-test, that there was no significant status, but as you can see in the center graph, there were signs of general proliferation in the HM group, which could indicate that the infant formula could be lacking some nutrients or some key factor that doesn't allow it to be as proliferative. And in the third graph, you can also see that there is more general or more proliferation of microglia in the IF group. And these dots indicate all the different groups that we use, HM1, 2, and 3, which indicate the piglets, for each respective set. And in order to quantify this and have greater significance with our next steps, we can look at more serology, we can add more piglets to the actual sets, and we can also do morphology studies. And you can see in this skeleton that we will be focusing on the branch points and comparing that with other microglia and using that to find the indicator of neuroinflammation. And we can also further investigate mechanisms of how diets affect neuroinflammation. And with that, I'd like to acknowledge the Weinstein Lab for the mentorship and the support, the Chen Lab for the collaboration, and the Piglet Brains the funding. And I'd also like to thank Dr. Ellenbogen, Ms. Ellenbogen, Julie and Sylvia, Dr. McDonald, Jim Pridgeon, and everyone who supported me in this program. Thank you.